Good day, listeners. My name is Professor Sunday Olajide Awofisayo. I am the principal researcher at Bioscird Research and Development Limited by Guarantee or Bioscird Research Group of Department of Clinical Pharmacy and Biopharmacy, University of Uyo. Today I'll be giving a lecture on clinical trials and public health. What are the learning objectives? We want to define clinical trials. We want to explain the relevance of clinical trials in public health. We want to also differentiate between the major types of clinical trials that we have. Phase 1, 2, 3, and 4. We want to explain the ethical principles that guide the use of humans in research. And we want to describe the historical development influencing modern clinical trials and the regulations that follow it. We want to also discuss the applicability of informed consent and participant protection in clinical trials. We will also look at the methods of conducting clinical trials. We want to analyze the randomized control trials, observational methods in clinical studies. We want to also look at the statistical and the epidemiological perspectives and approaches in clinical trials. We will also assess the public health significance of clinical trial outcomes in this one lecture of about 1 hour. Okay, we'll discuss the challenges and future direction in clinical trial research. We will also apply ethical and methodological principles in designing advanced clinical research studies. So let us go to the introduction. Clinical trials are fundamental scientific tools that we use to evaluate the safety, efficacy, effectiveness, and the quality of healthcare interventions. We have quite a number of interventions here and there that we hope will solve problems for us or make things better. Of course, there might be ways we've been doing things before, and somebody is coming up with an issue or an approach or a method, and so we're going to look at that as an intervention, and then statistically and scientifically look at how this can beat previous methods, and then we can adopt it. Right? So clinical trials form the bridge between laboratory discoveries and evidence-based healthcare delivery. Yes, there are quite a number of researches we've done, and we've come up with, oh, this can do this, this can do that. It could be ethanolic extracts of this leaf or this root or bark or stem, or it could be some molecular modifications, structural activity relationships, and then we've come up with a molecule that we think might be promising. There will be the need to, after we've gone through the preclinical methods, we want to, take it to the next level, having applied, and then we start looking at phase 1, phase 2, and phase 3. After approval, we also still look for the phase 4 of the intervention that we've done. So these are central to modern medicine and public health decision making and pharmaceutical innovations. So public health relies heavily on clinical trials because we want to prevent diseases, we want to improve therapeutic outcomes, we want to reduce morbidity and mortality, we want to develop evidence-based treatment guidelines. And so clinical trials will give us that evidence that this is efficacious and this is first safety, then efficacy, and superlatively better than what we've been having before. So that will give us improved healthcare approaches, and of course policies can be made on them. So clinical trials contribute immensely to development of drugs, vaccine evaluation, disease prevention, therapeutic monitoring, and healthcare policy formulation. So let us define what clinical trial is. This is a systematic scientific investigation involving humans and designed to evaluate the effects, safety, efficacy, pharmacokinetics, pharmacodynamics, or therapeutic value of a medical intervention. Of course, it might be drug, it might be some diagnostic approaches. So WHO defines clinical trials as any research that prospectively assigns humans to one or more health-related interventions so as to evaluate the effect on health outcomes. So what are characteristics of clinical trials? 1, human participation. 2, prospective design. 3, intervention assessment. 4, ethical approval. 5, scientific methodology. 6, define endpoints. 7, data collection and analysis. And the last one, controlled conditions. So what are the importance of clinical trials in public health? We want to generate evidence for healthcare decisions. We want to improve disease prevention strategies. We want to enhance patient safety. We want to support regulatory approval of medicines. We want to guide national treatment policies and improve healthcare quality. And finally, promote rational drug use. I remember sometime during the COVID-19, a man came around and says he has an extract from a leaf shown to him by the late mom, and he claims that can cure COVID-19 virus and the complications that go with it. And so, interrogating him, asking 1 or 2 or 3 questions, he couldn't just answer, and so that shows that he has not even started at all. And so we have examples of clinical trials, including COVID-19 vaccine trials, HIV and retroviral studies, malaria treatment evaluation, hypertension intervention studies. And in this lecture, we'll be talking more about some definite studies that has gone before now. So what are types of clinical trials? Based on the purpose of the clinical trial, we can have treatment trial, we can have prevention trial, there's also diagnostic trial, screening trial, quality of life trials. And so let's look at them one by one. Treatment trials that evaluate new drugs, and of course, with the phase 1 is the first time that drug or that intervention is being tried out in humans. So, for example, new anti-cancer drug evaluation. Okay, that is a treatment trial. And what about a prevention trial that assesses methods for preventing diseases? While the other one is talking about a therapy treatment, this one is talking about preventive, a kind of prophylaxis approach. And so vaccine studies, of course, when we give vaccines, we want to problems before they start. So maybe taking a rabies vaccine and all of that, those we have to pass through clinical trials. Then we have diagnostic trials. This evaluates diagnostic procedures. There are some methods or some reagents and similar stuffs that are designed to help diagnose diseases. And so that too can also pass through clinical trials. Example of that is cancer screening tests. So we want to be sure that that protocol can actually tell us if somebody has any of these common cancers that we grapple with today. Then we have some screening trials. These determine effective disease detection methods. Example of that is low dose CT scan versus chest X-ray, which is preferred. And finally, we have the quality of life trial, where we assess the comfort and quality of life of the participant and come to a conclusion that the intervention we were put forward is something that should be embraced by all. And of course, shouldn't forget to talk about behavioral trials. This one involves studying participants' behavioral outcome. This is a non-drug intervention, evaluating the counseling, education, habit change, or sometimes we use some digital tools. We want to see how this impacts on the participants so that we can now take those medical intervention or health intervention to the next level and becomes something that is used across the... And so example of behavioral trial include smoking cessation programs, and the primary endpoint of that is behavioral change. The fellow doesn't want to take a cigarette anymore and all of that. So let's move on. Classification based on study design. The first we mentioned were based on purpose. Now a classification of clinical trials based on study design. We have some randomized control trials, RCTs, and participants are assigned to this group or that group randomly. And so this forms the gold standard for testing in clinical trials. And so why we do a lot of randomized clinical trials is because it eliminates and minimizes biases, and the validity is very high because the persons who have brought into the work are not taken arbitrarily. So Because as we tell them to just say 1, 2, 1, 2, 1, 2, 1, 2. And so we have two groups already. Instead of just saying, okay, let us put this, we shouldn't be part of the, so that we don't introduce bias to the work. So the limitation of RCTs is that it is expensive and it is also time consuming. An example of RCT is Paradigm HF. That's a double-blind randomized clinical trial. Patients randomly assigned to groups. Non-randomized trials where patients are not brought into the available groups by chance. We actually meddle with how we put this person here or there. So investigators or participants choose the arm they are taking, whether this arm or that arm. Or we just allocate them to arm based on where are you from? Are you from this hospital, Urapa? Are you from that hospital, general hospital? Already, the work is already skilled. It's already looking as if the design is want to know the difference between this hospital and that hospital. Whereas we put the participants together and then we ask them to, we assign them randomly. Then there might be 27 of a Rapan staff here or patient here with a 32 of a general hospital, and this one is the other way. Unlike when we just use hospital-based, dividing them into groups. We want to also look at the way we blinded single blind, where the participants don't even know, they have information about the medical intervention. Then we have the double blind, where both the participant and the researcher are unaware of, especially if you are using a particular drug and a placebo. They don't even know which group is taking the placebo, which group is taking the real drug. And that appears to me as a better approach to remove biases and some confounding issues like that. Then we have the triple blind, where participants, the researcher, and then the analyst, those who are handling the data, don't even know which is which. They are just adding up values, finding mean, averages, and they're looking at the risk, errors, and all of that, without them even knowing what treatment happened to the groups. And so we also have placebo control trial, where the real drug, if it's a capsule and it's red in color, the placebo too is a capsule, red in color. The only difference is that there's no active ingredients in the placebo. And so this is administered to the participants, and we call that one a placebo controlled trial. And that's a gold standard for proving that a drug actually works. And because if somebody knows he's taking a placebo, and we're talking about headache, and you ask, is your headache still on? Yes, it seems that still have a little headache because he knew he was taking placebo. And so if we blind it, and then we also, so we can have a double-blind placebo control trial. And so sincerely, the clinical trial is an interesting research. And I remember that we're doing something like this in my laboratory in Unio about 3-4 years ago. And sincerely, it was very interesting. And so we have crossover trials where each person happens to be his own control. So having taken drug X, if that's the real drug, after some time we have a crossover, and the person is not taking the placebo or the other treatment. The same that took this other one will now be moved to the other group again, and at the end of the day we conclude the research where all the participants have had a touch of the treatment, whether treatment A or B. B can be placebo, or it may be something else that we are trying to evaluate. We may be looking at the comparative effectiveness of two drugs, A and B. And so A takes at this time this particular drug, B takes the other, and after the washout period we move them. And so these are very interesting touches. So example of a crossover trial that was done was that of methylphenidate that was taken for 2 weeks, and then placebo for 2 weeks, and then reversed after a washout period of 48 hours. And the research was N of 1 crossover for ADHD. Also a parallel group trial. This is the most common randomized clinical trial, where participants are randomized to a group or different treatment arms, at the start and remain there throughout the trial. So the participant stays in a group and does not get crossed to the other group, and whatever group some other participant had, they remain in that group throughout the length of and so randomization can be 1-to-1 or 2-to-1 or 1-to-1-to-1 if we have 3 treatments that we are evaluating. So the 1-to-1-to-1 stands for arm A, arm B, arm C, where arm A, B, and C are different treatments. Example of that is the paradigm heart failure control trial, where we have a double-blind randomized clinical trial of 8,442 patients. Okay, we move on to phases of clinical trial. We could have phase 1, phase 2, phase 3, phase 4. So look at phase 1. Phase 1 means that is the first time that drug or that intervention is being tried out in human. And of course, what we're looking at that point is safety. And they want to know the maximum dose that gives convenience, no serious issues here. So we are able to see the maximum tolerated dose at that phase 1. And healthy volunteers are used most of the time, except the disease we are looking at is cancer or maybe HIV and other more serious disorders. So if it is not so, then we're going to be needing healthy volunteers for the work. And this can last from days to months. And then the number of people that we bring into this are always from 20 to 100, not more than 100, precisely 20 to 100. Okay, so we'll be looking at the tolerability of that drug in these persons. Many clinical trials actually fail at that point when we look at the complaints and the devastation that that drug introduces to the participants. So in that phase 1, we also monitor the pharmacokinetic parameters of the drug absorption, whereas we look at the time that we achieve the Cmax, and then we look at the bioavailability of that drug at that initial. Then we also look at distribution parameters for that drug. What is the VD like for that drug? What is the protein binding profile for the drug? The metabolism, which of the CYP enzymes, or could it be the other enzymes in the liver that these drugs have to cope with in terms of the metabolism? Then how is the excretion? Is it renal? Is it hepatic? So with all of this, we are beginning to characterize the drug in terms of pharmacokinetics. There about the pharmacodynamic issues. We want to also know what the target steady state concentration is with regards to engagement. Does the drug actually hit the receptors, or is it actually impinging on enzymes? So what biomarkers are we going to be using to monitor the progress of levels of this drug? And if possible, if we are looking at cases like cancer, we want to also look at what biomarkers are we going to be able to say that this drug has actually done this or done that. And so we have a book already published by Bioscird Research Group, authored by my humble self, that expatiates on different biomarkers that a pharmacist, a clinician, indeed anybody that has to do with patient care has to read to get basic understanding of biomarkers. We look at the different organs. Is it the cardiovascular, respiratory, and all of that? And the book is about 35 chapters, simplified in every way for one to read through. Within 1 week, you are done with that book. You can read it again until you internalize the entire book. And so, example of phase 1 clinical trials include single ascending dose (SAD), where we have healthy volunteers get one dose, maybe 10 milligram or 30 milligram or 100 milligram, as we escalate the dose in cohorts, and they watch for adverse events. So in 10 milligram cohort, nothing, no serious adverse events. Then in 30, is there any? Then those who are taking 100, is there any? So those are approaches that helps us to know or ascertain the safety of this new drug that we claim can do wonders from in vitro or animal studies. Now that we're bringing them to humans, We want to be sure that what we anticipated is what we're getting. Right, so let's move on to phase 2. Phase 2, this is the first test of the drug in a patient. We have said before that phase 1 is healthy volunteers or participants, except for some particular critical conditions. Then phase 2 is the first time we're actually trying this out in people that have the disease for which this drug is being held out. So the main goal is efficacy here. And we're also still looking at safety. And because these sick persons, we still want to be sure that it does not complicate their issues. But here we have a larger participant number, 100 to 300. At this time, we've scaled up from 20 to 100. Now we're looking at 100 to 300. We don't use LD participants at this point. And the clinical trial can last from months to 2 years. So design is often randomized and may be blinded and may have a placebo. And single arm is common in oncology. Okay, the endpoints are surrogate or biomarker endpoints and not hard outcomes. In phase 2, we don't want to attend to these persons till death happens. Once we see some adverse events that are untoward, we discontinue and take a decision on the intervention that we are exploring. 3, this is also larger and is a more definitive trial. It proves that this drug actually works. In phase 3, we want to extend the efficacy profile that we saw in phase 2 to phase 3, and they want to be sure at this point that yes, this drug can actually cause a difference. And of course, we've already confirmed that the drug is safe. We further confirm that this drug is safe because they're still going to be monitoring for adverse events, side effects, and other stuff like that. So this is the point where Food and Drug Administration of America and the EMA, European Medicines Agency, confirms that this drug is what, given approval for marketing. About 300 to 3,000 people are used in phase 3. Sometimes for cardiovascular interventions, we might want to have up to 10,000 to 20,000 participants so we can be certain that the cardiovascular outcomes are real and what the approval that's about to be given. And this can last from 1 to 5 years. And so it's long enough to ascertain the endpoints that we have. And then we also appreciate the issue of endpoints that are hard here. For example, we want to find out whether this drug, adverse events, does it lead to death? How many people actually died during that body? So we can use that to say whether the drug should be given the approval or not. Remember that we said in phase 2, we don't want to administer the protocols hoping to record that 3 or 4 or 5 people died. We shouldn't even get to issues of deaths and all of that. But in phase 3, we accommodate that. We want to see how many people actually died. If nobody dies, oh great. But if people die, how many? And then let's statistically look at the benefits of that drug. The clinical endpoints for phase 3 include hard endpoints, including death, myocardial infarction, stroke, hospitalization, not just looking at biomarker. How did it change the biomarker? Is it troponin reduced or increased and all of that? Once did somebody actually give up the ghost? So phase 4 is a post-marketing surveillance. Most of the drugs on our shelves in our pharmacies or hospitals are supposed to be in that category now. There are some drug interactions that we didn't monitor in during the clinical trials. During this post-marketing, and I've done a lot of such phase 4 trials, and I'm sure it's been done here and there, having fulfilled all the protocols. We want to be sure that this drug does not interact with that drug, does not, and because sometimes drugs are not taken in isolation, and people have comorbidities and co-presenting diseases that will make a prescription put this drug with some other drugs, and this was not anticipated at the point of clinical trials. Use of humans in research. The use of human in research involves human participation in scientific investigations, generating biomedical or public health knowledge, And so humans may participate as healthy volunteers, they may participate as patients, they may participate as community population, and sometimes vulnerable groups. And when we talk about vulnerable groups, we want to exercise some strict protection for them. Let it not appear as if we are usurping or capitalizing on their vulnerability. So those counted as vulnerable include children, pregnant women, fetuses in the womb, prisoners, those who are cognitively impaired, elderly, economically or educationally disadvantaged, employees, their employers. So they're coming and enter. No. They are vulnerable because they don't want to lose their job. That's why. Okay. So students, critically ill minorities. So research using vulnerable patients. If research has potential benefit to them, then they can be included in that research. If extra protection is in place for them, they can also be used in that research. And of course, consent process must be in place. A parent permission, LAR, legally authorized representative of that person, especially in cases of those who have cognitive impairment, dementia, and all of that. There must be someone that the court says can give a yes for this person before such a person can be used in research. So briefly, we look at the historical background of human ethics. It will be okay to start with the Nazi human experiment, an experiment conducted during World War II, where persons, because they are prisoners, they're not free, they are vulnerable, were included in a research, and that led to this Nuremberg Code in 1947. And looking at the 10 principles that Nuremberg Code brought up as a result of the gross violation of human rights that happened during that World War II. So let's look at the Nuremberg Code very quickly. Number one, voluntary consent is absolutely essential. So if there's going to be any CT at this moment, then these people must say, yes, I'm willing, I'm ready, I want to be part of it. Okay? The experiment should yield fruitful results for the good of the society. It can't just be any research at all. Then number three, it must be based on animal experiment first, and knowledge of the disease that we are looking at. We must avoid all unnecessary physical and mental suffering that this research may bring. So we want to know this research that you want to do. What kind of pains? Are you going to be cutting people's skin, or are you going to barb their head, or are you going to bring some all those physical and mental sufferings? We want to evaluate and be sure that they are not there. Then it's not allowed if death or disabling outcomes are going to be recorded. Nuremberg says no, you cannot do that kind of research. The risk must not exceed humanitarian importance of the problem. Then we must protect subjects against even remote injury, disability, or death. The only scientifically qualified persons can conduct the research. I'm a technical secretary to the ethical committee in my school, and we see some persons apply for ethical approval, and you see the write-up does not show that these people can actually do a meaningful research. It appears that they're just wasting time and wasting resources and all of that. If people cannot dot their I's and cross their T's, and maybe the formatting 3.1 and they move to 3.4, where's 3.2, 3.3? So from the point of application, it shows that these people might not be able to do very thorough research. And if they have to gather people together and at the end of the day publish something that will mislead the so subjects can end participation anytime. And so you must let them understand that if you don't want to continue again, even tomorrow, after giving your consent now, you are free to opt out. Then scientists to stop experiment if it looks like we didn't know that with this kind of problems that can lead to disability or death. You must stop. You say, I know we have obtained the ethical approval, we must continue. No. You must stop. And so we also look at the one like we just discussed now, the Tuskegee syphilis study, where some medical doctors in America enlisted some persons for research without telling them what they're doing, and make them take drugs that can they actually even infected some participants with syphilis. and then started trying out 1 or 2 drugs on them. And because they didn't tell them, they only told them, actually, you have bad blood, come into this research, let us give you this drug. They deceived them. And some of them died. Some, having infected them with syphilis for the purpose of this research, some of them met with their wives and infected their wives with syphilis. And of course, you can see how messy all of that is. And that is why in the world today, you can't just do any research that involves humans, whether laboratory or even with questionnaires. A lot of persons are already aware that people have their rights to life, and so researchers cannot just bring them. So about 128 people died from that Tuskegee syphilis study. 40 wives were infected. 19 children were born with congenital syphilis. You can see the fracas, the mess that came with that. And so that led to Belmont Report 1979, where we now talk about respect for persons, beneficence, justice, and that also led to the establishment of IRB, Institutional Review Board, for ethical consideration of researches. And this must be in institutions where researches are done. Okay, so ethical principles in human research. The Belmont principle talks about the respect for persons, autonomy, informed consent, beneficence, justice, and the rest of it. Then this issue of informed consent has to do with ensuring that the participants give their informed written consent. If anybody goes contrary to that, it can attract some legal and heavy punishment. So, method of studying clinical trials, we're going to close with that. We've already talked about randomized clinical trials before, and then issues of randomization, control group, blinding, and comparing interventions. Okay, there are few trials. This is conducted in every population. Community trials, the entire community is studied. Observational methods involving cohorts, even within a group, and so we follow them over time, and then note what is happening to them, and then record the data from the observations, and then with that we can make valid conclusions. So randomization in clinical trials, it eliminates selection bias, improves comparability. Okay, and then we have simple randomization, block randomization, stratified randomization. And because of our time, we may not be able to explain how all of this play out, but we can look at how these randomizations can be done all in the RCT design. Control groups can be placebo control, active control, or historical control, and the purpose is for good comparative evaluation between the interventions that we have on ground. And data collection, we adopt this method: clinical observation, laboratory investigation, questionnaires, interviews, and electronic data systems for our data collection. Validity and reliability, we have internal validity, accuracy within study, external validity, generalizability, and reliability, consistency of results. So these are what we consider in all the data that we've put together. So what are the biases? We have some selection bias when unequal participant selection are made. We have observer bias, researchers influence on the entire work, and then we have recall bias, participants memory error. They say, I can't remember. Did you do this? I can't remember. These are some errors introduced into the work. Attrition bias, some persons don't even show up again. They actually had high spirit at the beginning of the work, but before you know it, they're no longer coming up. So what are the confounding parameters? Confounders distort association between exposure and outcome. And so how do we control this? By randomization, matching, restriction, and statistical adjustment of our data to take care of the confounding factors. So what are the statistical methods in clinical trials? That's where we're going to stop this afternoon. Important concepts: hypothesis testing, p-value, confidence interval, relative risk or risk ratio, odds ratio. These are all that we look at as we put our data together. So public health application of clinical trials. It helps in developing vaccines, improve maternal health, control infectious diseases, reduce chronic disease burden, improve national health care policies. Distinguished ladies and gentlemen, we will close by talking about some challenges of clinical trials. High cost. Yes, you can imagine running a research for months, not to talk of years. And so some costs go into millions of dollars. And that is why most of these clinical trials are sponsored by developed countries, though they may be done in developing countries. Rarely do you find the funding for that starting or emanating from developing countries and ending there. So future directions are as we've listed on this slide. Thank you for listening.